Hi guys, this is Miss Bastido here to give us an introduction to the topic of evolution. So this will be a pretty short video. We'll have a separate video over microevolution and macroevolution. So as we start this section, we just need to sort of focus on some definitions that we need to know. So that's what we're going to do first. And so there are three sort of definitions that you should be able to describe. And first one is you should be able to define evolution. And so you can read what's written here, but the idea is that this is a change in the genetics of a population over long periods of time or over multiple generations, and that this could result in the development of a new species. The key here is that it might not, but it could. And so as we see these genetic changes over really long periods of time, that could lead to some sort of isolation, and that isolating mechanism could then lead to reproductive isolating and then eventually a new species. When we talk about microevolution, which is probably what you're most familiar with, these are small scale changes. So really, when we're talking about natural selection, typically within a population and those small changes that occur, this occurs within the same taxonomic group or the same species typically. And so, if we have, for instance, black beetles and tan beetles, and the tan ones are eaten by the predator, and so they die out over time. Those black ones changing, they're still the same species, but the species has changed a bit over time. We call that microevolution. And that really microevolution we'll have a whole video about, but it is mostly driven by natural selection, but could be genetic drift or something like that as well. With macroevolution, now we have variation in allele frequencies above the species level. And so the changes are so much that now we have a formation of a new species or a formation of a new taxonomic group. And so the descendants then are different enough to be classified as a different species, and we call that macroevolution. And so here's a good example of microevolution. And so we have these peppered moths, as they're called. And these peppered moths actually, this is before the Industrial Revolution. There was a lot of lichen growing on the tree, and you might remember that lichen grows on trees where there's nice, good air quality. And so the lichen is very light colored, and so over time most of the moths were this light color. There were occasionally dark ones, but you can tell the light one blends in a bit better here. And as during the Industrial Revolution, this is mostly in Europe, but during the Industrial Revolution, what happened is this light colored lichen stopped being produced as much and growing because the air pollution had gotten bad enough in the area that the lichen wasn't growing on the tree. And that caused the bark on the tree, the dark bark on the tree, to be exposed. And so you can see that what would happen over time here is that before the Industrial Revolution, we found that most trees had this light-colored lichen on it, and the phenotype that was preferred in the environment would be this light-colored moth. And so these black ones would often get eaten by birds because they weren't camouflaged. And then what we found was that over time, after the Industrial Revolution, the lichen wasn't growing on the trees anymore, that the variation in the population changed and that there was more dark-colored moths because now their variation or their alleles that allowed them to be dark-colored actually became an advantage. And so they survived better. And if they survive better, they're able to reproduce and pass on that trait for being dark-colored. And so over time, the allele frequencies in the population would change to more this darker color. They're now finding the opposite to be true again, as the air pollution is not quite as bad anymore. And so the opposite has now happened again, which is that the light-colored moth has now been preferred. This is microevolution, and microevolution because we haven't formed a new species. We've just changed the alleles within the population, and so the genetics within that population are changing, but in terms of forming a new species or a new taxonomic group, that has not happened. And when we talk about macroevolution, we're really talking about new species forming over time. And so when we look at this cladogram or phylogenetic tree, we see that all of these species share a common ancestor here. And we'll look at another one of these in a minute, but all of them share a common ancestor here. And so they all have this trait, which is they all have vertebrae. So these are all vertebrates. And then the sharks are evolving here. Then we have a bony skeleton comes. And so then the fish, everybody above this level has a bony skeleton. Everybody above this level has 4 limbs. Everyone here has an amniotic egg. Everybody here with eggs with shells. And so you can kind of see that hair sort of evolved here. And so macroevolution is really that the formation, this is a common ancestor, but then at some point a bony skeleton came into existence, and that allowed for the formation of a new species. Now, this didn't happen overnight, likely, that there were many small changes that occurred, and then those small changes eventually lead to some sort of isolating mechanism. And so some kind of reproductive isolation has to occur in order for this to form a new species. You should also be able to look at evolutionary timeline and be able to identify points in time where there was large amounts of evolutionary radiation, but also mass extinctions. And so evolutionary radiations, we're talking about really large increases in numbers of species. And so if we just look at this graph, we can see this is the number of families, and that goes back to, this isn't species, it's talking about a larger taxonomic group than that, but still equivalent to number of species, except for number of species would be even higher. And so here, anytime you have a big increase, we would say that is a point of evolutionary radiation. So between the Cambrian and we call that the Cambrian explosion, that we have a really large increase in species. And then we kind of stays the same for a while, goes down a few times. We'll get there in a minute. And then here we have during that Jurassic period, we have a really large number of species increase. And again, here in this period here. And then again, in the Tertiary period, we have a big increase. So these are all times in evolutionary time that we've had huge increases in the number of species or evolutionary radiation. And then also, how can we identify mass extinctions? Well, then we're looking for decreases. And so, especially sharp decreases. So in this case, they're identified with the dot, the mass extinctions. Some of those are a little less obvious on this graph, but you can see them a little bit better here. So in this graph here, instead of graphing the number of families, they've done the opposite and graphed the rate of extinction. So anytime you have a big peak, that would be a rate of extinction. And so you can see these different peaks. And basically they said if a certain percentage have gone extinct, then this is a mass extinction if we've gotten above that line. And so these mass extinctions we currently have 6 mass extinctions, and we're currently in the 6th. So there have been 5 mass extinctions through time. With the most recent one being the dinosaurs after the Jurassic period, or actually after the Cretaceous period, but before the Tertiary period that we had that mass extinction of dinosaurs. And now we're currently, it's thought to be, into the 6th mass extinction. The difference here is now this mass extinction is almost exclusively caused by human activity. So whether it be deforestation or pollution or climate change or lots of other things. And so this mass extinction is human caused, where these mass extinctions were caused by other events that happened on Earth. So on the exam, you might see one of these timelines and have to be able to identify these sorts of things. And so the last part here in this section, in this just intro video, is just a little bit about how to create a phylogenetic tree. And so I'm going to quickly go over it. So if you look here, we look at the number of amino acids difference in cytochrome C. Cytochrome C is just a protein that most species produce. And so these are different species of, I have no idea what. And these species, it'll show the number of amino acids that they have different from each other. And so the more differences they are, the further away they should be on the table. And so that's the first thing I look for, is who has the most differences. And we see Ephorus and Polylepis have the most differences. And so we would put them the furthest apart. And so we've put Polylepis here, and that's because it's also quite different from everyone else in general. So this one is quite different from all of them. So we put them as the outgroup. It is the most different from everyone. And that means Ephorus is the most because it's the furthest away from that, because it's 21, we put it on the other end. And then we look, then I would start from this side and say, who's the most closely related to E. ferus? And so we can go to E. ferus and see that Africanus is the most closely related. And so we would put a line here for Africanus. And then we have the other two. And so you have Gallus and Fosteri. And Gallus is more closely related to Ferus than Fosteri. And so you can see that's 11 and 13. So we need to put Gallus closer to Ferus and then Fosteri from there. Remember that these lines represent time. And so we want the lines to be equal here and here, especially if both of these species are still alive. And so if I asked you, where is the most recent common ancestor between Africanus and Fosteri, you should be able to identify that. And so I'll give you a second to look it up. Who is the most recent common ancestor between Fosteri and Africanus here? And so your answer should be here. And so that's the point at which we have a common ancestor. That's the first place where they both would meet, would be here. Now, if I asked you between Gallus and Fosteri, then it would be here. They have a common ancestor here with each other. And there's only one common ancestor for everyone, and that's farther down here, and that would be further back in time. So hopefully, this should have been, phylogenetic trees are really simple to the cladograms we did in Unit 3, and so hopefully those are not too difficult for you to figure out. But you should be able to, you might not have to build one, but you should be able to interpret one in terms of, they could give you something like this and then show you 4 different trees and ask you which one would be correct. And so just understanding you're always looking for the ones that are the most different, put them furthest apart, and then work from there. Hopefully you found this video helpful. And we only have 2 more videos left in Unit 4, so that will be one over microevolution and one over macroevolution. Let me know if you have questions.